Okay, so welcome everyone to the 54th lecture of Dr. Heidery Step 1. We apologize for starting the lecture a bit late today, after half an hour. We were going through some issues regarding the weather once again. I hope you guys are doing good. Can you guys hear my voice? Can you guys hear my voice? First and foremost, let me make sure you guys can hear my voice. Okay. So once again, let me apologize for starting the lecture a little bit late. We're having another storm over here. And what happens that during the storms, it gets a little bit difficult to connect a bit earlier in the mornings, and that's the issue. So that's that. But hopefully we could start by 9:30. Um, so instead of wasting time and waiting for people to come and all the students to join, do you want to begin the process of revision and recapitulation from yesterday's lecture? Is everyone ready to begin the revision and recapitulation of yesterday's lecture? Okay, very well, very good. So let's start with the revision. First of all, we talked about mutations in DNA, and we have two types of mutations, single nucleotides and other types of mutations. If we have a mutation where there is no change in the amino acid, what type of mutation is this? Very good. At which position is the mutation taking place? Okay, good. At the third position. Okay, very good. Next one is if we have a mutation at which a nucleotide substitution changes the entire amino acid, what type of mutation is this? Okay, if we have a nucleotide substitution which results in an early presence of very good early presence of a stop codon, it's a nonsense mutation. If there's a mutation which cannot be divided by the number 3, what type of mutation is this? Frame shift mutation. Very good. If there's a mutation which results in inability to remove the introns, what type of mutation is this? Splice site mutation. Okay. Next one is uh, in the environment of E. coli, in the environment of E. coli, if there is low glucose and high lactose, what would happen to the lac operon? Activated. Okay. What is the mechanism of activation of lac operon by low glucose? How does low glucose activate the lac operon? Okay. How does high lactose activate the lac operon? Okay. What is an example of a nucleotide excision repair defect? Okay. What is the example of a base excision repair defect? Good. What is the example of a mismatch repair defect? Is my voice breaking or is the voice okay? Do I have a, okay, good. Breaking. If my voice is breaking for you, Dr. Sabira, I think it's better that you refresh the meeting and I'll let you in again. Don't worry about it. For everyone else, is the voice okay? Okay. Next one. Next one is what is an example of non-homologous end joining DNA repair defect? Ataxia. Okay, good. What is the example for homologous recombination defect? Okay, good. Then the next one is if we have um, if we want to increase the genetic transcription, at which point or at which part of gene expression do we activate the regulators, the promoters? Very good. Okay. What are the names of the two promoters? What are the names of the two promoters in the genes? Cat box and Tata box. Very good. Okay. What is the process of post transcriptional modification? Capping, trimming, 5-methyl cap. Okay. Poly A tail. Very good. Okay. And the splicing out of introns. Okay. So splicing. Okay. What are the ribosomes that are synthesized from RNA polymerase 1? Okay. What are the ribosomes synthesized from RNA polymerase 2? Okay. What are the ribosomes synthesized from RNA polymerase 3? Okay. Okay. Does the RNA polymerase have proofreading abilities? Okay. What type of proteins are required to form a spliceosome? Which proteins are required to synthesize or to form a spliceosome? U1 or U2, S, N, R, and P. Very good. Okay. Out of, if we mix match the exons after removing all the introns so that we can form different types of proteins, what is the name for this mechanism? The alternative splicing. Okay, good. Okay. If you visualize a tRNA, what is the function of the T arm of the tRNA? Binds to the ribosome. What is the function of the D arm? D arm, D. Detect. Okay, it binds to the tRNA. Okay. Then what are the 3 nucleotides that are present at the amino acyl binding end? CCA. Okay, good. Okay. Next one is protein synthesis. And from protein synthesis, we don't really have a lot of questions for you. Can you guys name the eukaryotic ribosomes? Eukaryotic ribosomes. Okay. Okay, very well, very good. Okay, so having said that, are we confident from the lecture from yesterday that if we receive questions from those parts, we can knock them out of the park? We can give really good answers? Yes. Okay, good. Very good. How about everyone else? How do you feel in terms of your confidence regarding the first portion of biochemistry? The genetic portion? Because right now we will start with the cellular portion. Okay, good. Okay, so let's begin with the cellular portion of biochemistry. And the first thing that we're going to discuss today is, okay, the first thing that we are going to discuss today is the biochemistry cellular, that is the cell cycle phases, cell cycle phases. Cell cycle phases, basically, these are checkpoints which controls the transitions between the phases of the cell cycle. That is, we talked about the cell cycle, how in the cell cycle you have the M phase, then the G phase, then the S phase, and then the G2 phase. So cell cycle phases, these are the checkpoints between the phases. These process is regulated by cyclins, which are proteins, cyclin dependent kinases, that is proteins with phosphates, and tumor suppressors. So you basically have three things: cyclin, cyclin dependent kinases, and tumor suppressors. These are the three things basically which are responsible for controlling the phases of your cell cycle. These are actually pretty high yield, so I need you guys to write it down separately over here so that it's better when you guys do a revision. You guys can quickly look at through this. Okay, this is high yields. You only need three things: cyclin, cyclin dependent kinases, and tumor suppressors. M phase, which is this phase over here. This is the shortest phase of the cell cycle, which includes mitosis, that consists of prophase, prometaphase, metaphase, anaphase, and telophase. And we know these phases, we have studied them before. So that's that. After that, what we have is we have cytokinesis. Cytokinesis is the process where the cytoplasm, it splits into two. And the G1 and G0 are of variable duration. That is, G1 phase and the G0 phase could be of variable duration. So mitosis is basically the process of division, right? Mitosis, that is, you have metaphase, prometaphase, anaphase, telophase, right? And then cytokinesis is when the cytoplasm, it divides, it goes into two directions on the action of the spindles. And then you have G1 and G0, which could be of variable phases. That's that. Now let's talk a little bit about these three things, that is cyclin, cyclin dependent kinases, and tumor suppressors, that is the regulators. Okay. So number one is cyclin dependent kinase. Number one is cyclin dependent kinases. They are constitutively expressed but inactive when not bound to cyclin. So basically, they have to be bound. Cyclin dependent kinases, when they are not bound to cyclin, they are inactive. They are not working. Then you have cyclins. Cyclins are regulatory proteins that control the cell cycle. They are phase specific, and they activate the cyclin dependent kinases. Basically, cyclins also do not work at their maximum potential if they are not bound to cyclin dependent kinases. So these two, they have to bind together. So when they bind together, the structure they form is called cyclin dependent kinase complex. Cyclin dependent kinase complex. The action of these complexes are that they are responsible for phosphorylation of other proteins to coordinate the cell cycle progression. So they will phosphorylate, meaning that they will add kinases. The word kinase means they can add the phosphate group. So they will phosphorylate. What happens is when you phosphorylate another protein, this can either activate or this could inactivate the protein for the cell cycle to progress. So depending on the type of protein this is, this will result in activation or inactivation. The questions you will get from over here is they will ask you the name of this one over here. They will ask you the name. That is, first of all, they will give you a question, and then they will have answer options for you. For example, A, B, C, D, and E. Over there, you can have cyclin dependent kinase, cyclin dependent kinase complex, tumor suppressors, or something else, right? Something else. Over there, they will ask you which of this is responsible for the addition of phosphate, and most of the student will write cyclin dependent kinase. But you have to realize that cyclin dependent kinase itself will not act until and unless it's bound to the cyclin. So A and B are wrong answers. C is the right answer because cyclin dependent kinase, this is the one that is responsible for the addition of the phosphate. So this is your right answer. Okay, so keep that in mind. Next one is tumor suppressors. Tumor suppressors are basically, there are P53, and then there's P21. P53 will activate P21, which will activate cyclin-dependent kinase inhibition. What happens is when you inhibit cyclin-dependent kinases over there, what happens is that the cell cycle progression stops, and this results in RB hypophosphorylation or RB activation. RB is another type of tumor suppressors. So if you inhibit CDK, RB gets activated, and when RB gets activated, this results in G1 to S phase progression inhibition. This results to G1, this is your G1 over here, G1 to S phase progression is inhibited when RB is activated. RB is a tumor suppressor gene. RB is a tumor suppressor gene, right? So that's that. Mutations in tumor suppressor genes can result in unrestrained cellular division. So you have Li-Fraumeni syndrome, which is a very high yield syndrome. Over here, the patients have a family history, and they also have a history of having multiple carcinomas at the same time. And there's another one that is growth factors. For example, insulin, platelet-derived growth factor, erythropoietin, epidermal growth factor. They will bind to the tyrosine kinase receptors for the transition the cell from G1 to S phase. Okay, so RB is responsible for inhibition, and growth factors are responsible for activation. So what did we learn from over here is that the cell cycle phases, we learned about the regulators. We learned about CDK, cyclin, CDK-cyclin complex, and tumor suppressors. Over there, we discussed one question already from the UWorld, that is, which of this is responsible for the addition of phosphate, and the answer is CDK-cyclin complex, because CDK itself is not active. Cyclin itself will not work to its maximum potential until and unless it's bound to CDK. Then we talked about tumor suppressors. Number one tumor suppressor is P53. If it gets activated, it will activate P21. P21 will inhibit cyclin-dependent kinase. This will activate RB, and RB will prevent the transition from G1 to S phase. That's that. And the exact opposite way is the working of the growth factors. Growth factors will bind to the tyrosine kinase receptors for the transitions of the cell from G1 to S phase. Okay. So next time you guys receive another question regarding the tumor suppressor genes, and they ask you at which phase of the cell cycle is the tumor suppressor gene P53 responsible for working, and your answer should be from G1 to S phase at this phase of the cell cycle. Does anyone have any question? Does anyone have any question? Okay. Yes, what is your question? What is your question? Please, we do not have a lot of time. Please let me know your question so that I can answer it for you. RB is a tumor suppressor gene which will prevent the transition from the G1 to S phase if it gets activated. Okay, that's the action of RB. Can we move forward? Can we get some yes and no to understand whether we should move forward or not move forward? Okay, thank you so much. Good. Next one. Next one are the cell types. The cell types. How many types of cell are there? The types of cell that are usually there, they are permanent. Okay, they are permanent, stable, and labile cells. Okay. So first one is permanent. Okay, permanent cells. What do we mean by permanent cells? Permanent cells are the cells, permanent cells. These are the cells that will remain in the G0 phase, that is in this phase. That is in this phase, G0 phase. The advantage of staying in a G0 phase is that they can be activated whenever you need them to, meaning that they can be regenerated from the stem cell. So basically, if something stays at G0, easier for them to regenerate and form the new cells. And the cells which are responsible for staying at the permanent cells are N, S, C, R. NSCR, that is one second, New York Sports Club royalty. New York Sports Club royalty. N for neurons. N for neurons. S for skeletal muscle. C for cardiac muscle. R for RBC. New York Sports Club is it's basically a gym, right? What happens when you go to a gym is that you can let your neurons fire, right? Your neurons are firing when you are working out. You are working on your muscle, you're working on your skeletal muscle, then your heart rate goes up, you're doing cardios, so cardiac muscle. And royalty is that after you work out, you feel like a royalty. So RBC. So neurons, skeletal, cardiac, RBC. Did you guys write them down? Did you guys write these down? Yes or no? Okay. Okay. Okay, one second. New York Sports Club royalty. New York Sports Club is a gym. Helps you. So what happens is when you work out in the gym, your neurons fire. So neurons. S for skeletal muscle. C for cardiac muscle. R for RBC. Okay. You are welcome. Okay. How many of us know a guy who is extremely stable in their life, meaning that he is not very crazy, that he is not the type of a person who would go out every night, have fun, go to parties, be with different types of people. What he likes to do is he likes to stay home, listen to poetry, or eats pastries. Do we know any guys like this who are very stable guys? Does anyone know any stable guy like this? Who's not really into going out a lot, likes to stay home, right? So we know about a stable guy. You know who our stable guy is? Our stable guy is this guy over here, okay? What he likes to do is he's very stable in his life right now, right? So what he does, so he's a very stable guy. At this point in his life, he has done his fair share of everything. So right now, what he does is he likes to stay home and eat pastry and learn poetry. So he, meaning John, likes pastry and poetry. So he's very stable. He stays home all day, likes to read poetry and eat pastry. He likes pastry and poetry. This is for a stable John. Okay, H4, hepatocytes. L4, lymphocytes. P4, proximal convoluted tubules. And P4, periosteal cells. Okay, H for hepatocytes, L for lymphocytes, P for proximal convoluted cells, and P for periosteal cells. Okay, are we clear about this? You guys write this down? Can you spell again the mnemonic? The mnemonic is stable. He likes pastry and poetry. Pastry. Okay, next one. Next one is, okay. Before we move on to the next one, wait for a minute. The thing about this, the stable cells, the stable cells are, what happens is that the permanent cells, they remain in the G0 phase. The stable cells, they remain in the G1 phase. In the G1 phase. So they stay over here in the G1 phase. And when they are activated, only then they enter the G1 phase from the G0 phase. But until and unless they are not activated, they will remain in the G0 phase with the permanent cells. But the hepatocytes and the lymphocytes and the proximal convoluted cells and the periosteal cells, they can enter the G1 phase very fast. So whenever they're activated, they can enter the G1 phase, and then they can help in cellular regeneration. That's that. Okay. Another one is this this is known as labile cells. Labile cells, and the thing about the labile cells are you do not need to have any other mnemonic for the labile cells, because every other mnemonic that is used for these ones, that is neuron, skeletal muscle, cardiac muscle, and RBC, and hepatocytes, lymphocytes, proximal convoluted cells, and periosteal cells, every other cell in the body that are not these cells are labile cells, meaning that everything else. So you have your bones, bone, especially the bone marrow, right? You have skin, then you have germ cells, then you have hair cells, hair, right? And then you have gut epithelium. Okay, so these are the cells that are not the permanent or the stable cells, and these are the labile cells. The problem with these cells are that the labile cells, they never go to the resting phase. The resting phase is the G0 phase, so they never go to the resting phase, right? So they keep on dividing rapidly, and their phase in G1 is very low. So what happens is that these are the cells that are extremely affected by chemotherapy. You know how chemotherapy, what they do is they halt the cell cycle progression. They halt the cell cycle progression from here to here, and we have studied them in our oncology drugs, right? We have a lot of drugs who work in this cell cycle over here. So the chemotherapeutic drugs, if you are asked about a question that which type of cells are most affected by chemotherapy, the answer is the labile cells. And if they ask you why, then your answer is that they never enter the G0 phase, and they divide rapidly with a very short G1 phase. That's why. So labile cells are your answer. That's that. And with that being said, we have talked everything about the cell types. Okay, so we have talked about the cell types. Okay, can I give you guys 1 minute? Okay, I'll give you guys 1 minute to name all the cells. Okay. It's 10. I'll ask you the question at exactly 10:01. Okay. So what are the permanent cells? What are the cells which are in the permanent cell type? Can you guys name the mnemonic, please? What is the mnemonic? Very good. New York Sports Club royalty. So, neuron, skeletal muscle, cardiac muscle, and RBC. What are the cell types that are, well, what are the example of the stable cells? What is the mnemonic? What is the mnemonic? Okay. He likes pastry and poetry. Ratio, and the rest are the labile cells. Which type of cell types are the most affected by chemotherapy? Okay. Okay, good, very good. Next one. Next one that we want to discuss is rough endoplasmic reticulum. Rough endoplasmic reticulums, these are basically organelles, that is, they are present in the cytoplasm. Rough endoplasmic reticulum are the site of synthesis of secretory proteins. So the proteins which are secreted are synthesized over here. This is a high yield term, secretory protein. So if you are asked which organelle is responsible for the synthesis of secretory proteins, your answer is rough endoplasmic reticulum. So they are responsible for synthesis of secretory proteins and also of N-linked oligosaccharide addition to lysosomal and other proteins. So they are also responsible for synthesis of lysosomal proteins, secretory proteins, out of which secretory protein is more high yield, and oligosaccharide with an nitrogen-linked terminal end. That's that. Then there are things that is Nasal bodies. Nasal bodies are rough endoplasmic reticulum in neurons. So basically, Nasal bodies, we have talked about this before. Do you guys remember our discussion on Nasal bodies? Do you guys remember our discussion on Nasal bodies? Yes or no? Nasal bodies, when we were talking about Nasal bodies, where did we talk about Nasal bodies? In the neurons, in physiology. Do you remember? In neuron regeneration and neuron damage. Exactly. We talked about Nasal bodies. The Nasal bodies are rough endoplasmic reticulum, which are responsible for the synthesis of peptide neurotransmitter for secretion. So once again, neurotransmitters are a type of protein, and they are for example, the neurotransmitters are which are responsible to be secreted are synthesized by the rough endoplasmic reticulum. So once again, secretory proteins are being synthesized. Next one is free ribosomes. Free ribosomes, which are unattached to any membrane. These are the site of synthesis of cytosolic, peroxisomal, and mitochondrial proteins. Cytosolic, peroxisomal. Peroxisomal meaning that the proteins which are synthesized for the peroxisomes. Peroxisomal and mitochondrial proteins. So these are the site of synthesis of these proteins. That. Basically, the best way to remember this is that rough endoplasmic reticulum is responsible for the synthesis of secretory and lysosomal, and everything else is synthesized in the free ribosomes. That. Mucus secreting goblet cells of the small intestine and antibody secreting plasma cells are rich in rough endoplasmic reticulum. So basically, write down over here that if any cell requiring secretion, any cell requiring secretion should be high in rough endoplasmic reticulum, because secretory proteins are synthesized by the rough endoplasmic reticulum, and these proteins are then released. So any cell which is responsible for secretion should be high in rough endoplasmic reticulums. Proteins within the organelles, example, endoplasmic reticulum, Golgi bodies, lysosomes are formed in the rough endoplasmic reticulum. This is not that very high yield. The only piece of information you need to take from over here is the secretory proteins and how they are synthesized by the rough endoplasmic reticulum. Next one is smooth endoplasmic reticulum. We all know what the function of this one is, and this is very high yield. The function of smooth endoplasmic reticulum is that this is the site of steroid synthesis and detoxification of drugs and poisons. So basically, steroids are synthesized in the smooth endoplasmic reticulum and detoxified, and the drugs and poisons are detoxified over here. They lack surface ribosomes. That's why they are smooth. They do not have any ribosomes. And the location of glucose 6 phosphatase is also smooth endoplasmic reticulum. So we will talk about this later. But The only question that is high yield from over here is what is the site of steroid synthesis? And the answer is smooth endoplasmic reticulum. This is very high yield. Liver hepatocytes and steroid hormones that are responsible for producing the cells of the adrenal cortex. So we know that the adrenal cortex, they're responsible for the production of a lot of high yield steroids. So thus they should be extremely rich in smooth endoplasmic reticulum. So once again, any cell responsible for increased steroid synthesis should be high in smooth endoplasmic reticulum. Smooth endoplasmic reticulum. Okay, write it down over here so that it's easy for you to review the next time you read first aid. Okay. Next one is cell trafficking. Are we all clear about the first page? Are we all clear about this, about the first page? Okay. Which type of organelles are responsible for secretions? Which type of organelles are responsible for drugs and toxin detoxification? Okay, good. Next one is cell trafficking. Cell trafficking. Cell trafficking is basically Golgi bodies, or Golgi is the distribution center for proteins and lipids from the endoplasmic reticulum to the vesicles and plasma membrane. So all the proteins that are synthesized, secretory proteins, steroid, or any other proteins that are synthesized by the smooth endoplasmic reticulum, after they're synthesized, they are not released outside. What they do is they send these proteins to the Golgi bodies for post-protein formation and modification. So for the post-translational modification that happens in Golgi, they include what are the translational modification. I want you guys to write this down over here, and I'll do it on a blank page because this is high yield. The reason why I ask you guys to write things over here is that next time when you guys read first aid, you guys can go through your own notes on the side so that it's easy instead of going through over here. So you don't know exactly if you're missing out on anything if you have stuffs and notes written over here. So basically, what are the post-translational events or modifications that happens in the Golgi apparatus? First of all, the first one that happens is if you have a protein that is there in the Golgi body, and what happens is if the, like how proteins are made from a lot of different types of amino acids, yes or no? There is aspartate, glutamate, aspargine, right? Am I correct? A lot of different types of amino acids are required to form a protein because a lot of genetic codes are there, and each genetic code codes for a specific type of amino acid. Am I correct? Yes or no? Am I correct? Okay, good. So what happens is the first process of Golgi apparatus is that they add or they modify the N-oligosaccharide, my apologies, N-oligosaccharide on aspargine. Aspargine is an amino acid. So they modify the N-oligosaccharide on aspargine. Basically, they modify the oligosaccharide with the nitrogen N on aspargine. Okay, so that's that. Next one is if they find that the protein has serine and threonine, what they do is that they add O-oligosaccharide to serine and threonine. Okay, so these are the first two functions. Next one is extremely high yield. This one is the last function of the Golgi apparatus, and this is very, very high yield. That is, they add mannose 6-phosphate to the proteins, to all the proteins, regardless if they have serine, threonine, asparagine. This is one function of Golgi apparatus, which will happen regardless of anything else. That is, they will never forget to add the mannose-6-phosphate to the proteins so that those proteins can be transferred to the lysosome. If you do not add mannose-6-phosphate, then these proteins cannot go to the lysosomes. Lysosomes are another organelles, right? And if the proteins, they do not go to the lysosomes, those proteins, they cannot be matured properly. As a result, those proteins will stay inactive or immature or premature. So in order for the proteins to go to lysosome properly, the Golgi apparatus will bind a mannose-6-phosphate to the proteins. So these are the three functions, and these are the functions of Golgi apparatus. I want you to write down over here. N terminal N to asparagine, O oligosaccharide to serine and threonine, and mannose-6 to the proteins for lysosomal trafficking. I know it's written over here. I just want you guys to write this one shortly over here so that you guys can next time view this, because this is the only piece of information from all these things that is extremely high yield. And next to adding of mannose-6-phosphate, put your stars. Okay. Next one. Next one is endosomes are the sorting centers for material from outside the cell or from the Golgi and sending it to the lysosome for destruction or back to the membrane. So basically you have another organelle that is endosomes. Endosomes, basically what they do is endosomes are present inside the cell, and when they receive a protein, they either send it to lysosome for destruction or they send it to the Golgi body for modification. That's that. So if anyone asks you what is the mechanism of endosomes, basically they are the sorting centers. They sort whether the protein is a good protein. If it's a good protein, they will send it to the Golgi. If it's a bad protein, they will send it to the lysosome for destruction. That's that. Okay. Okay. Is everyone clear about this? Is everyone clear about this? Okay. So what was the most important function of Golgi bodies, which I just said? What is the most important function of the Golgi bodies, which I just mentioned? Very good. Addition of mannose-6-phosphate to the proteins. And what happens if we add the mannose-6-phosphate to the proteins? What happens? What happens if we add the mannose-6-phosphate to the proteins? Transfer to lysosome. Okay. So what happens is, what if I tell you that there is a disease, okay? There is a disease which is basically a failure of the Golgi apparatus or the Golgi bodies. The Golgi bodies, they fail to add the mannose-6-phosphate to the proteins. As a result, the proteins are not delivered to the lysosomes. They are delivered straight outside of the cell instead of going to the lysosome. If the proteins are not delivered to the lysosomes, are those proteins mature proteins or premature proteins? Premature proteins. Okay, very good. Okay, so those proteins are premature proteins. This disease is known as Isil disease. Isil disease. Isil disease is extremely high yield. Okay, especially for the fact that most of the time it's difficult to identify Isil disease because it's more or less a lysosomal storage disorder, right? But the clinical features of this one, they overlap with a lot of other clinical features. So what happens is, if we draw a patient with Isil disease, okay, if we draw a patient with Isil disease patients, first of all, they have very coarse facial features. Coarse facial features mean very prominent facial features like this one, coarse facial features. That is extremely prominent, like a very prominent nose, prominent eyes, prominent lips. So like that one. So coarse facial features. So if we draw big eyes, big nose, big lips, you know what? Let me use another one. Okay. So this is for coarse facial features, Isil disease. Next one is these patients, they have clouding of the cornea. So basically clouding of the cornea, if, okay, over here, but everything else is clouded, okay? There's clouding of the cornea. Corneal clouding. Do you guys know another lysosomal storage disorder where there's corneal clouding? Okay, corneal clouding. Do you guys know another lysosomal storage disorder where there is corneal clouding? Very good. Ehlers syndrome. Okay. Okay, good. Next one. Next one is another one that is this one over here. This one is for gingival hyperplasia. These patients, they also have gingival hyperplasia. Okay, gingival hyperplasia. Then what happens is, let's come to their hands. Over here, they have restricted joint movements. Okay, restriction of joint movements, all because the proteins could not be transported to the lysosomes. So every other protein in the body is affected, restricted joint movement and big claw hands or claw hands, right? Basically, claw hands. Along with this, some patients can also have kyphoscoliosis, okay, where they bend forward, right? So they can have bent out back or kyphoscoliosis, okay? Kyphoscoliosis. So these are the clinical features of Isil disease. Okay, first of all, corneal clouding, then coarse facial features, gingival hyperplasia, restricted joint movement, claw hands, and kyphoscoliosis. Okay. Okay. Is this a scary picture? Yes or no? Looks pretty scary, doesn't it? It's pretty scary, doesn't it? Okay, good. So that's that. Okay. So this is your Isil disease. Let me show you a picture of the Isil disease. Isil disease. Okay. This is Isil disease. It's a lysosomal storage disorder. Very unfortunate, but that's that. Okay. So are we clear about this? Isil disease. Now let's look at the text. Let's see what the text says. So Isil disease is basically this is known as inclusion cell disease or mucolipidosis type 2. Mucolipidosis type 2. Okay. This is an inherited lysosomal storage disorder where there's a defect in N-acetylglucosamine L phosphotransferase. This results in failure of the Golgi to phosphorylate mannose. So the reason why Golgi bodies cannot phosphorylate or add the mannose to the proteins for lysosomal trafficking is because there's a defect in an enzyme. The enzyme is basically a phosphotransferase enzyme. So you don't have to remember N-acetylglucosamine L. All you have to do is you have to remember that the mannose 6-phosphate cannot be added because in the Golgi, there is a deficiency or there's a problem with phosphotransferase. As a result, the proteins are secreted extracellularly rather than delivered to the lysosomes. This results in the coarse facial features, gingival hyperplasia, clouded cornea, restricted joint movement, claw hands, kyphoscoliosis, and also there's another one that we missed out, that is high plasma levels of lysosomal enzymes. So if you do a serum study, you will see that the lysosomal enzymes are high over there. This is often fatal in childhood. Okay, most of the time, the mortality is so high that unfortunately, the children, they usually do not survive. So that's that. This is fatal. Okay. So are we clear about Isil disease? Are we clear about Isil disease? Can we identify Isil disease? Can you guys tell me really quick what are the clinical features of Isil disease before we move forward? Coarse facial features. Then what else? Corneal clouding. Then what else? Gingival hyperplasia, restricted joint movement. Okay. Claw hand, kyphoscoliosis. Very good. So these are the Isil disease findings. Okay, next one. High serum lysosomal enzyme. Okay, next one. Next one is signal recognition particle, or SRP, signal recognition particle. These are basically abundant cytosolic ribonucleoprotein, that is the ribonucleoproteins that are present in the cytoplasm, that traffics a polypeptide ribosome complex from the cytosol to the rough endoplasmic reticulum. These are abundant cytosolic proteins that traffics the polypeptide from the cytosol to the rough endoplasmic reticulum, meaning that they take it from the cytoplasm to the rough endoplasmic reticulum. If there is an absent or dysfunctional signal recognition particle, this will result in accumulation of the proteins in the cytoplasm. Very easy to understand. That is, you have some proteins. Okay, these proteins are responsible for transferring the polypeptide ribosome complex, right, from the cytosol to the rough endoplasmic reticulum. If there is no protein like that, then the proteins would accumulate in the cytoplasm. This piece of information over here, signal recognition particle, these are not very high yield. Okay, so you do not really have to focus a lot on this because there's not really a lot of questions that will come from over here. Next one is vesicular trafficking proteins. Vesicular trafficking proteins are basically, you have COP1, COP2, okay, and you have Clathrin. That's that. So COP1, okay, COP1 are basically these proteins over here. Okay, these are responsible for the Golgi to Golgi transfer, Golgi to Golgi, or Golgi to endoplasmic reticulum transfer. So vesicular trafficking proteins are basically, so you have COP1 over here that are responsible for trafficking the proteins that from the Golgi, that is from this Golgi to this Golgi, that is one, and then from this Golgi to this one, that is rough endoplasmic reticulum. So if you are asked, which you won't be because this is also not very high yield, but for any reason, if you are asked a question, what is the name of the protein which transfers the proteins from the Golgi bodies to the endoplasmic reticulum, the answer is COP1. COP2 is responsible for transferring the proteins from endoplasmic reticulum to the Golgis. That is in this way over here. And is in this way over here. This is for COP2. COP2. COP2 is responsible for anterograde, that is movement from the rough endoplasmic reticulum to the Golgi bodies. That is that. And next one is Clathrin. Clathrin is trans-Golgi to lysosomes. So Clathrins are these ones over here. They are responsible for transferring from the Golgi to the lysosomes. So this way. Golgi to the lysosomes. Very easy to understand. And then from the lysosome, you go to the endosomes, and that's that. So if I ask you very quick, which type of vesicular trafficking protein is responsible for transferring from the endoplasmic reticulum to the Golgi apparatus? What is your answer? Endoplasmic reticulum to the Golgi. What is your answer? Okay. Which one is responsible for trafficking between Golgi to Golgi? Okay. Which one is responsible for trafficking the proteins between Golgi to lysosomes? Okay, Clathrin. Very good. Okay, very good. Next one. Next one is, to be honest, a very, very favorite organelle of mine. Okay. This one is a very favorite organelle of mine, that is peroxisome. I don't know why I feel so attached to this organelle, and maybe because of the function, I'm not sure, but especially I love organelle because you can have so many questions from this organelle, because dysfunctions of peroxisome can result in a lot of different types of types of diseases which are very rare, which we are not aware of. But unfortunately, these diseases are very well tested in your exam. So peroxisomes are extremely high yield. First of all, let's understand what a peroxisome is. Peroxisome is a membrane enclosed organelle. What does that mean? That means that the organelles are enclosed within the membrane. And the functions of peroxisomes are very high yield. That is, they are responsible for beta oxidation of long chain fatty acid, very long chain fatty acid, alpha oxidation of branch chain fatty acid. Okay, these two are strictly peroxisomal processes. That is, basically, if there is a long chain fatty acid, very long chain, and branch chain, then beta oxidation of very long chain occurs in the peroxisome. Alpha oxidation of branch chain also occurs in the peroxisome. Next one is catabolism of amino acids and ethanol. If there is a catabolism of amino acids and ethanol, that occurs in the peroxisome. That is the breakdown. Next one is synthesis of cholesterol, bile acids, and plasmalogens. Plasmalogens are basically important membranes of phospholipid, especially in white matter of the brain. But if there are two things which you need to focus on, that is alpha and beta. Well, what does that mean? That means that peroxisome is responsible for alpha oxidation of branch chain fatty acid, and it is responsible for beta oxidation of very long chain fatty acid. These two are extremely high yield, especially for peroxisome, so alpha and beta. Next one is, what are the diseases that can occur in a patient of peroxismal disorder? That is, the diseases are Zellweger Syndrome, Refsum disease, and adrenoleukodystrophy. So let's talk about these diseases in a blank page. Okay. What I need you guys to do is, if you guys have post-it notes, okay, if you guys have post-it notes, you know, like how you have post-it notes which you can attach to this page over here, so that there's a small piece of information on first aid, and then you can turn your pages easily to find the peroxismal disease. On that post-it note, write down rare disease, and then post it over here and keep it hanging out of the page, so that every day when you get the chance, you can see the peroxismal diseases at least once, because this will be really difficult for you to keep in mind. And the only way that you can remember these diseases is by re-exposing yourself to the text again and again. Okay. Now, so the first one that we want to talk about is Zellweger syndrome. Zellweger syndrome. Okay. Zellweger's is basically, the reason is because over here in the peroxisome, there's a mutation in a gene. The mutation gene is PEG's gene. So the cause is pathogenesis, is mutation of PEG's gene, P-E-X, PEG's. As a result, these patients, what do they have? Okay. What do they have? First of all, they have seizures. Then they have, if I try to draw the muscles of the, then these are floppy muscles. By floppy muscles, what do I mean? These are not taut muscles. These are floppy muscles. By that, I mean that these patients, they have hypotonia, floppy muscles. The tone is not normal, so hypotonia. So seizures, hypotonia. Then over here, you have hepatomegaly. Okay, these are the three things. And then of course, there's another one, that is death. But to be honest, death is not a clinical feature, and you will not receive questions about this, that you have a dead patient. So seizures, hypotonia, and hepatomegaly. These are the three ones. Okay, and this is Zellweger syndrome. This is one of the peroxismal disorder. Over there, the mutation is in a gene which is responsible for proper peroxismal function, and that gene is known as PEG's gene. This is the pathology, and the causes are seizures, hypotonia, and hepatomegaly. Okay, if you guys want, you guys can draw this diagram so that it's easy for you to understand. Next one is Refsum disease. Refsum disease. Okay, Refsum disease is also high yield. Refsum disease, or as I like to call this disease, not Refsum. I like to call this disease rough skin disease. This is my mnemonic for Refsum disease. Why do I like to call this rough skin disease? This is because over here we talked about the fact how peroxisome is responsible for alpha oxidation of branch chain fatty acids. So what happens is, if there's no proper alpha oxidation of the branch chain fatty acid, we have fatty acids, for example, phytanic acid. Okay, phytanic acids are branch chain acids, and if they are not broken down, they are broken down. So we have, let me write it down for you. So we have phytanic acid, and these are broken down to pristanic acid, pristanic acid. So phytanic acid is a branch chain fatty acid, and it's broken down to a pristanic acid. If this does not happen, what happens is, if this process does not take place properly, what happens is you have a patient, for example, these patients, do you remember how I called Refsum disease rough skin disease, right? So these patients, they have scaly skin. This is the number one finding: dry, scaly, rough skin. Scaly skin. Along with this, these patients, they have cataracts. Okay, cataracts, night blindness, and that's that. And then they also have another one. This is not very high yield, but still, I really have no idea the reason behind this one. But apparently, if they have all toes over here, 1, 2, 3, 4, 5, the fourth toe is small, short fourth toe. Okay, that's that. So this is basically Refsum disease. And the treatment for Refsum disease is basically you have to fix the diet. That is, you cannot give these patients extremely branch chain fatty acid, or you have to supply them with proper pristanic acid supplementations. And this is that. Either you fix the diet, or there's another one, that is, in treatment, what you can do is you can do plasmapheresis. Plasmapheresis, they provide with all new types of proteins, and that can help with another type of peroxisome, which can actually work in a better way. So you have two treatments, that is diet and plasmapheresis. So this is basically Refsum disease or rough skin disease. Okay, rough skin. Okay. Are you guys drawing it down along with me? Are you guys drawing it down, drawing down the mnemonics? Okay, because if you don't draw these down, it doesn't matter how many times you read this. It does not matter how many times you read these things. You can never retain this knowledge until and unless you have some sort of a drawing or anything which will allow you to visualize the whole thing. If you can visualize it, then you can easily retain the information. Okay, next one is adrenoleukodystrophy. Adrenoleukodystrophy, if I have to categorize the diseases according to the importance, then adrenoleukodystrophy is number one. Refsum disease is number 2, and Zellweger syndrome is number 3, according to the importance. Adrenoleukodystrophy is number 1. So adrenoleukodystrophy, first of all, if you have improper activation, improper functioning of the alpha oxidation, then you get Refsum disease or rough skin disease. And if you get improper activation of the beta oxidation of the very long chain fatty acid, then you get adrenoleukodystrophy. Adrenoleukodystrophy. Okay. So what happens is that these patients, they are X-linked recessive. X-linked recessive. By the way, patients of Refsum disease are autosomal recessive. Both of them are autosomal recessive, except adrenoleukodystrophy, which is X-linked recessive. Okay, they are X-linked recessive disease. X-linked recessive disease. And the pathology over here is that these patients, they do not have proper beta oxidation of very long chain fatty acid. They do not have the proper beta oxidation of very long chain fatty acid. Why? Because they also have a mutation. The mutation is an ABCD1 gene. Zellweger syndrome was due to the mutation of PEG's gene, and adrenoleukodystrophy is due to the mutation of ABCD1. Okay, as a result, if the very long chain fatty acids which are responsible to be broken down, if they cannot be broken down, this results in buildup. So very long chain fatty acid, they are not broken down in the peroxisome, so they are building up. So results in, okay, they are building up in where? They are building up in the, one second. Okay, they are building up in the brain since they are not broken down. Then where? Okay, they are building up in the brain. Then they are building up in the, not the kidneys. Okay, not the kidneys, but something on top of the kidney, that is this one over here, adrenals. So that's why it's called adrenoleukodystrophy, right? So adrenal, so brain, adrenal glands, and another one is gonads or testis. Okay. As a result, these patients, they can actually die from adrenal gland crisis. They can die from adrenal gland crisis because the adrenal glands are responsible for secretion of stress hormones, that is cortisol, and those are extremely high yield. So in a point of stress, for example, during an operation or anything else, if we do not get enough cortisol, the patient can actually die. So these patients can die from adrenal gland crisis. But this is adrenoleukodystrophy. Okay. Are we clear about this? It's a washout. Okay. Okay. Can I give you guys 2 minutes? I'll give you guys 2 minutes to read peroxisome. What I will ask you is, let me tell you what I will ask you. I will ask you the functions of peroxisome. I will ask you the names of the 3 diseases of peroxisome. I will ask you the genetic predisposition of the peroxisome. I will ask you the clinical features of each of those 3 diseases. And that's what I will ask you. So I'll give you guys 2 minutes on the clock. Okay, 3 minutes. Okay, 3 minutes. I'll give you guys 3 minutes. Because the reason is I want you guys to read and master first aid right now during the lecture, so that the rest of the day can be spent on doing questions. So that's done. Okay, 3 minutes. Okay. Is anyone ready? If they are, please let me know so that I can ask them questions and active participation. Okay. So one student. I would need the help of at least 3 students who can help me out. 2. Another 1. Anyone else? 3. Very. Okay. 4 students. Okay. Good. So let me ask you guys the question. First question goes out to Dr. Mohammed. If you would be willing to unmute yourself, that's okay. If you want to use the chat box, that's okay. It's up to you. My first question is basically peroxisome. What are the functions of peroxisome?
Alpha oxidation of branch amino acid, beta oxidation
That's 1. Then
long fatty, very long fatty amino acids, catabolism of amino acids, and ethanol, synthesis of cholesterol, bile acid, and
did you
plasma.
mention the catabolism of amino acids and ethanol?
Yes.
Okay, you did. Very good. And you mentioned beta oxidations of very long chain fatty acids and alpha oxidation of branch chain fatty acids, right?
Yes.
Okay, very good. Thank you. Thank you so much, Dr. Mohammed. And the
Thank
last one you kept on saying, but let me finish it for you. It's synthesis of cholesterol, bile acids, and plasmalogens. Okay. So are we clear about this, Dr. Mohammed? The functions of
Yes,
peroxisome?
yes, thank you.
Okay, very good. Thank you. Thank you so much for participating. Next one goes to Dr. Iman. Would you be kind enough to tell us the clinical features of Zellweger syndrome?
Seizure, hypotonia.
Okay. Hypotonia, seizure, and another one. Do you remember the drawing of the big liver?
Yeah.
Hepatomegaly. Okay, very good. Thank you so much. Next question goes out to Dr. Hussein, Dr. Samra Hussein. What is the pathogenesis of Zellweger syndrome? What is the gene that is mutated in Zellweger syndrome? Okay, the question goes to Dr. Mridula. What gene is mutated in Zellweger syndrome?
PEX gene.
PEX gene, very good. Okay, and is anyone else, okay, Dr. Mridula, would you be kind enough to tell us what are the clinical features of Refsum disease or rough skin disease?
Scaly skin.
Scaly skin.
Cataracts and night blindness.
Cataracts and night blindness.
And shortening of the fourth toe.
Okay, shortening of the fourth toe. Very good. Next one is, I think I forgot to mention one other sign, that is, in Refsum disease, the patients have some degree of ataxia. Ataxia, okay? Ataxia basically meaning that these patients are also affected in the sense that they have an ataxic gait, okay? They have an ataxic gait. Okay, next one is Dr. Samra Hussein. Would you be kind enough to use the chat box and provide the answer if the voice is not working? What are the clinical features of adrenal leukodystrophy?
Clinical feature of
okay, not the clinical features. Where do the very long chain fatty acids build up? Well, what
accumulated, yeah, in the brain, adrenal gland, and testes.
Very good. Thank you so much. That's that. So hope everyone understood the questions and that. And next time, if you guys receive questions from Peroxisome, would you be confident or not confident to answer them? Yes or no? Okay, good. Next one is proteasome. Proteasome. Okay, proteasome. These are barrel-shaped protein complexes that degrades damaged or ubiquitin tagged proteins. So ubiquitin is a type of protein, so what happens is when you tag a protein with ubiquitin, it goes straight to the proteasome, and then there's destruction of that protein. Do you guys remember how we talked about the fact that in cachexia, do you guys remember that we talked about this, and you all know that in cachexia, you have muscle breakdown because they're tagged by the ubiquitins, and then they go to the proteasome to be degraded? Yes or no? Who remembers that? Right, we talked about this. So ubiquitin, if you tag a protein with this substance, this will go directly to the proteasome, and then this will get degraded. If there's a defect in the ubiquitin proteasomal system, defects in the ubiquitin proteasomal system have been implicated in some cases of Parkinson's disease. That is, in Parkinson's disease, some proteasomes, they do not work. The only thing over here you have to understand is if you tag something with ubiquitin, they will go to the proteasome. That's that, nothing else. No other question. Next one is cytoskeletal elements. Now we will talk about cytoskeletal elements. These are basically a network of protein fibers within the cytoplasm that it will either support the cell structure or help with movement and cellular divisions. I'm sure we are all very well aware and well acquainted with these types of filaments. That is, over here we have microfilaments, intermediate filaments, and microtubules. Microfilaments are basically, okay, microfilaments are actin and microvilli. These are responsible for muscular contraction and cytokinesis. Basically, microfilaments, we all know what actin and myosin do, right? That is, they are responsible for muscular contraction and also cytokinesis, that is this division of the cytoplasm, which we talked about. Next one is intermediate filaments. Intermediate filaments are responsible for the maintenance of the cellular structure. Intermediate filaments are vimentin, desmin, cytokeratin, lamins, GFAP, and neurofilaments. I'll tell you in 1 second how you can remember everything. Just let me finish this. Next one is microtubules. Microtubules are responsible for movement and cellular division. First of all, microfilaments, easy to understand that it's muscle contraction and cytokinesis. Intermediate filaments are intermediary structure that will maintain the cell. Microtubules are responsible for movement. So we know microfilaments, if it's responsible for muscular contraction, it's actin. If microtubules are responsible for movement, then of course the example should be cilia, flagella, mitotic spindle, and mitotic spindle. Axonal trafficking centrioles are not that high yield, but cilia, flagella, and mitotic spindle. So if actin is microfilament and cilia, flagella, and spindles are microtubules, everything else, every other filament is an intermediary or intermediate filament. So if you receive a question in your UWorld that which structure is desmin, then I would like you to think of the fact that desmin, okay, if you cannot remember what desmin is, try to remember that you have filaments such as actin and myosin, which are obviously not intermediary filaments, they are microfilaments. Then try to think about, you have movement filaments, example, cilia, flagella, right? And those are your microtubules. So obviously desmin, vimentin, and all the other ones, they are intermediary filaments, okay? I will give you guys exactly 1 minute to learn this, and then I'll move forward. Okay, I'll give you guys exactly 1 minute to learn this, and then I'll move forward. What I will ask you is the examples for each of the different types of the filaments, that's that, and the functions, okay? So 1 minute on the clock, it's 10:51. I will ask you at 10:52. It's very easy to understand. Okay. So what are the examples of microfilaments? Very good. What are the examples of microtubules? Cilia, flagella, and another one. No, not the centrioles. Centrioles are not very high yield, although centrioles are there, but the answer I'm looking for is mitotic spindle. Okay, these are microtubules. And everything else, what type of filaments are they? Such as desmin, vimentin, GFAP, lamins. These are intermediate filaments. Very good. These are intermediary filaments. Okay, next one. Next one is microtubules. Microtubules, which are responsible for movement and cellular divisions, we will learn about the structures of the microtubules. Microtubules, they have a cylindrical structure, which are composed of a helical array of polymerized heterodimers. That is, it's in a helix like this. Okay, the microtubules are like a helix. Okay, so you have one, and then you have another one like this. So this is the helical structure. They are a helical array of polymerized heterodimers of alpha and beta tubulin. Okay, so each dimer they have two GTP bound, has two GTP bound to them. They're incorporated into the flagella, cilia, and the mitotic spindle. So these are the three structures where microtubules are there. They will grow slowly, collapse quickly, and they're also involved in slow exoplasmic transport in the neurons. The molecular motor proteins, they are responsible for transport of cellular cargo towards the opposite end of the microtubules, meaning that there are some molecular proteins, right? So we have dynein, and then we have kinesins. So dynein and kinesins, they are not microtubules. What they are is they are molecular motor proteins. So these are proteins. So the thing is, with the help of the microtubules, dyneins and kinesins will transport a substance either retrograde or anterograde. Retrograde meaning retrograde meaning that if for example, this example over here, that is Clostridium tetani, herpes simplex, and poliovirus, they will use the dynein for retrograde transport to the neuronal cell. That is, they will transport the virus retrograde to the cell body, meaning that from the periphery to the central, right? And anterograde is kinesin. Kinesin will transport from the cell body to the periphery. Okay, so for example, we have a lot of different types of diseases. For example, Clostridium tetani, herpes, polio, rabies. They are viruses which affects the spinal, which affects the segments of the spinal cord. So in order for them to be transported from the periphery, which they have entered, to the central spinal cord, they have to go retrograde. Retrograde means going backwards. So they will use the dynein ones. And if there's anything which wants to go anterograde, for example, neurotransmitters, they can use kinesin, anterograde. So that's that. Okay. Is everyone clear about this? Retrograde and anterograde. Next time, if you have a microbiological question, which molecular motor protein are they using in order to be transported to the neuronal cell body? What should be your answer? What should be your answer? Molecular motor protein are they using in order to be transported? The answer is dynein. Okay, that dynein is a molecular motor protein. It's not a microtubule. Okay, anterograde is going forward from the cell body to the periphery. Anterograde is going forward from the cell body to the periphery. Repeat. Okay, so basically you have two types of molecular motor protein. One is dynein, another one is kinesin. Dynein is responsible for transporting something from the periphery. Let's say that this is the human, okay? This is the spinal cord, right? And this is the periphery. If you have any virus which wants to be transported from the periphery to the central spinal cord, that is towards the cell bodies, then they will use the molecular motor protein that is dynein, this way, dynein. And if anything wants to be transported from the central to the periphery, then they will use kinesin. That is another molecular motor protein. That's all I'm saying. So dynein and kinesin. Okay. Next one that we want to talk about is some drugs that will indefinitely attack the microtubules. What are the names of some drugs that attacks the microtubules? The drugs are microtubules get constructed very poorly. Microtubules get constructed very poorly, and the mnemonic for first aid is absolutely excellent, because this mnemonic will help you realize that you have drugs such as mebendazole, griseofulvin, colchicine, vincristine, vinblastine, and paclitaxel. These drugs attack especially the microtubules. Out of this, all of this, we have studied vincristine, vinblastine, and we have also studied paclitaxel in oncology drugs. So that's that. But microtubules get constructed very poorly. This is a very high yield mnemonic, so don't forget this. Okay, are you guys clear about this? Can I give you guys 1 minute? I'll give you guys 1 minute to understand microtubules get constructed very, very poorly. just read this and I'll ask you the names of the drugs that affects the microtubules. Okay, names of the drugs that affects the microtubules. Then I'll move on to over here. Okay. 1 minute on the clock, it's 10:58. I'll ask you exactly at 10:59. Okay, is anyone ready? Okay, what are the drugs that affect the microtubules? Indazole, Risofalvin. Then microtubules get constructed colchicine, then Verifor, Vinblastine, then paclitaxel. Very poorly, paclitaxel. Okay, very good. That's that. So these are the drugs that are used for microtubules. Then next one is structures of the cilia. Okay, so next one is structures of the cilia. Okay, ciliary structure. Okay, so have you guys understood the lecture till now properly? Yes? Okay, good, very good. Okay, so let's talk about the ciliary structure. Okay. Ciliary structure, I want to talk about this on a blank page over here. Okay. Ciliary structure, they have a very special type of organization. Okay, they have a very special type of organization. What they have is, if I try to draw a ciliary structure like this, how does a cilia look like? So let's say that this is the ciliary structure. The cilia, they have 9 doublets. Okay, let me just draw this very fast. So they have 9 doublets. Okay, so doublets meaning that they have like this one, 2, 3, 4, 5, and then there's 6, almost done, 7, 8, and 9. So something like that. They have 9 doublets. So basically the doublets, okay, the doublets, they are microtubule A. They are microtubule A, and there's microtubule B. In microtubule A and microtubule B, you have the diamond arms. So this microtubule A and B, they form a doublet, and there's 9 of them. 1, 2, 3, 4, 5, 6, 7, 8, 9. And in the central, they have another singlet. I mean 2 singlets. Okay, so they have 2 singlets in the middle. They have another microtubule, and then they have another microtubule. So next time if they ask you how many microtubules are there, okay, so you have 9 pairs and then two other more. So that's that. Okay, and each of the microtubule, that is microtubule A and microtubule B, they are connected by each other by another protein. That is this protein over here. This protein in the middle. This protein is called nexine. This is not a high yield protein at all. You will not be tested on this, but just for understanding the structure, because I know a lot of us will go into research, and this is important. These knowledges are important for your research studies. So nexine. Nexine is the protein that is responsible for holding microtubule A and microtubule B together. Okay. And so what happens is, over here, the dianine arms, that is this arm over here, that is the dianine arms, The ciliary structure, we know that the cilia, if for example, the cilia that is there in the respiratory tract, they move back and forth, right? So the cilia, if there's a cilia, it will move in this way, and then it will move in this way to make sure that the mucus that is being secreted from the goblet cells, it goes and moves up and up or down, especially up towards the esophagus, from where it could either be swallowed or sneezed out or coughed out. So cilia has to move. And the way the cilia moves is that the dining arms over here, okay, the dining arms over here, the axonal, they have an ATPase. They have an ATPase, and these ATPase, it links one pair with another pair. That is the dining arm. They allow for the pairing of one pair of A and B to another pair of A and B. So when the ATPase breaks down the ATP to ADP and PI, this results in activation of the dining arms, and the dining arm can form a bridge like this and pull on this one. And this results in movement of the entire ciliary structure, either in this way, or if it pulls, since you have on both sides, and if it pulls on that way, then the cilia can move into that way. So this is basically the movement of the ciliary structure. This is how the cilia moves. So basically, if we do not have the dining arms working properly, then the cilia will not move. If the cilia doesn't move, then what happens is a lot of the movements of the bodies will stop. For example, mucus secretions will stop, sperm motility will stop. Then there's another thing. There are gap junctions between these ones. There are gap junctions between each of the cilia. For example, this cilia is there, then there's another cilia over here, right? There's another cilia over here. So there's gap junction over in between one cilia to another cilia, right? Like this one. So when this one moves, this one activates the dining of this one, and then this one moves together. That's how you have coordinated movement of the cilia. It's not like one cilia is moving, another cilia is not moving. It doesn't work like that. Every cilia in the body moves in a coordinated manner. And the way it happens is because there is a gap junction, which results in the transport of impulses. So that's that. That's how the dining arms are functioning. So if we do not have this type of proper ciliary function, can we name a disease that is very high yield that can take place if we do not have these type of ciliary movements? What is the name of that disease? Cartagena syndrome. Very good. What is the another name for this disease? Another name for this disease. Primary ciliary dyskinase, and that is the only highest yield thing that you have to understand from the cilia. The even more important than the structure is the pathology of the cilia. Okay, so we'll talk about Cartagena syndrome. Cartagena syndrome. Before we talk about Cartagena syndrome, let's talk a little bit about this portion. That is the cilia structure we talked about. They have a 9 doublet and 2 singular arrangement of microtubules, which we just said. The basal body, that is the base of the cilium below the cell membrane, they consist of 9 microtubule triplets with no central microtubule that is in the base, not at the top. Okay. Axonal dining are ATPase that links the peripheral 9 doublets, and they cause the bending of the cilium by differential sliding of the doublet. So we just talked about this, how the dining arms, right? They're responsible for movement. And the gap junctions between one cilia and another cilia will enable coordinated ciliary movement, coordinated movement. So this might be a question. If there's uncoordinated movement of the cilia, where is the issue? Is the issue in dining or gap junctions? The issue is in gap junctions. If there is no movement, then the issue is in dining. That's that. Now let's talk about Cartagena syndrome. Cartagena syndrome is primary ciliary dyskinesia. What is the pathology of Cartagena syndrome? The pathology of Cartagena syndrome is you have immotile cilia. The cilia is not moving. If the cilia is not moving, then obviously the defect is in the dining arm, because the axonal dining is responsible for back and forth movement of the cilia. So defect in dining arm. Defect in dining arm. Okay. So what happens is, if you do not have proper movement of the cilia, what would happen? First of all, First of all, let's talk from the very basic. That is, when a heart has been formed, the heart is. Did you guys know that when the heart is formed, the heart initially is on the right side, and then there's cardiac looping, and then the heart loops with the help of the Diane arm. It moves from the right side to the left side. We talked about this in cardiac embryology. No? Okay. So basically, when the heart is formed, the heart stays, especially on the right side, right? And then with the help of the ciliary contraction, the heart, it moves from the right side to the left side to its normal position, that where the apex is at the left fifth intercostal space, right below the nipples. That's the normal position of the heart. But in order for the heart to come, what happens is initially, the heart is actually like this. Okay, the heart is actually like this. And then with the help of the cilia, right, the heart moves in this way. And so the final position of the heart is like this. It loops, it moves. Okay, so this is the process. So right now, if the cilia are not functional in a patient, what is the position of the heart in those patients? Can we say that these patients, they have dextrocardia, meaning that the heart is on their right side? Okay, so clinical features, if we have to talk about clinical features, then these patients, they can have dextrocardia. Dextrocardia. Okay. Then what else? They can have, okay, so we talked about the heart. Now we want to talk about the lungs. What happens in the lungs? In the lungs, these patients, they have mucus accumulations, right? And mucus accumulation results in necrotizing infections, and this can result in destructions of the cartilage, and as a result, there can be extreme dilatation. This is known as bronchiectasis. There could be bronchiectasis. Along with this, these patients could also have, you have sinuses. Sinuses, they contain mucus, right? And those mucuses, they have to be drained properly, or else you will have sinusitis. Then the mucus in the air, if that is not drained in the ears, then you can have otitis. Okay, then what else? Then if you do not have proper movement of the sperm, or you do not have proper movement of the cilia in the fallopian tube, you will get infertility. Infertility. Okay, the difference between infertility in primary ciliary dyskinesia and cystic fibrosis is, in infertility in primary ciliary dyskinesia, you have immotility and immotile movement of the sperm and the cilia of the fallopian tube. In cystic fibrosis, you have absence of vas. That's why in cystic fibrosis, you have infertility. Okay. Next one is that these patients, they can be diagnosed by nasal nitric oxide test. There's a test known as nasal nitric oxide test, which is not high yield, but nasal nitric oxide test can diagnose the Cartagena syndrome. Okay. Has everyone understood the clinical features? Has everyone understood the clinical features? Yes, if there is cystic fibrosis in women, there would also be infertility. Okay, so this is known as Cartagena syndrome. Now let's read the text. Okay, now let's read the text. First of all, Cartagena syndrome is immotile cilia due to Diane arm defect. It is an autosomal recessive syndrome which results in decreased male and female fertility due to immotile sperm and dysfunctional fallopian tube. There's increased risk of ectopic pregnancy over here because as you can realize that in the female fallopian tube, after you have fertilization in the fallopian tubes in the ampulla, then you have its movement to the uterus. So if that doesn't happen and the cilia are not working, then risk of ectopic pregnancy, that is the fertilized product will stay over there for a long time. So that's that. The next one is bronchiectasis. This can cause bronchiectasis because, for obvious reasons, the mucosal infection, sinusitis, otitis, conductive hearing loss, and situs inversus or dextrocardia. That's what we just say. And we can use nitric oxide, nasal nitric oxide, to see whether the patient has primary ciliary dyskinesia or not. Okay, are we clear about this? Okay, are we clear about this? How dynein arms connect to other microtubules. Dynein arms do not connect to other microtubules. Dynein arms are responsible for moving the microtubules A and B left and right, and the cilias, they are connected to another cilia by gap junctions. Okay, are we clear? Cystic fibrosis will result in absence of vas deferens in case of males. In case of females, it will cause mucosal secretions which are extremely thick. That's how your patients, they will have extremely thick mucus. That's why they cannot have proper pregnancy. Okay, that's that. Okay, next question. Gap junctions, what do they do with the dynein? Gap junctions result in coordinated transport of the impulses from one cilia to another cilia so that there is coordinated ciliary movement. That's why there is gap junction. Any other question? Axonemal dynein. Axonemal dynein are the dyneins that we have just talked about over here. Where were they? These are axonemal dynein. They use ATPase to break down ATP into ADP and PI, and then this results in the release of energy that causes the movement of the microtubule A and the microtubule B. Okay. Any other question? Any other question? Okay. Okay, let's move forward. Okay. This is a picture of dextrocardia, by the way. So you see how the heart is like this, whereas the heart should be on this side. Okay. This is very high yield. Next one is sodium potassium pump. What is a sodium potassium pump? We all know what a sodium potassium pump is. Can you guys name one hormone which results in increased formation of sodium potassium pump? And as a result, if that hormone doesn't work, the sodium potassium pump formation will decrease and patient will get lethargic. Nope. Nope. Thyroid. Very good. The thyroid hormone. Thyroid hormone. We talked about this in endocrinology, how thyroid increases the basal metabolic rate by increasing the activity of the sodium potassium pump. Do you guys remember that? You guys remember how we talked about this, that the thyroid, they increase the basal metabolic rate by increasing the sodium potassium pump activity. Okay, so that's that. The sodium potassium ATPase, it is located in the plasma membrane with ATP site on the cytosolic side. Okay, so if we have a sodium potassium ATP, I'm not going to use any more diagrams because this is a very good diagram over here. So as you can see that this is a sodium potassium pump and the ATP side is on the cytosolic side. So this is the cytosome, this is the cytosol, this is the extracellular space. Over here you have the ATP side, that is the ATP is broken down to ADP, and this results in the release of a phosphate. So what happens is that the channel opens up, and as a result, 3 sodium leaves and 2 potassium enter. So potassiums have a higher concentration outside than inside, and sodiums have a higher concentration inside and outside. So they have a tendency of leaving, and the potassiums have a tendency of entering. So plasma membrane is an asymmetric lipid bilayer containing cholesterol, phospholipids, sphingolipids, glycolipids, and protein. Not high yield at all. Don't worry about this. Pumpkin is basically the pump the potassium in. The potassium will pump in. So sodium potassium pump will 3 sodium out, 2 potassium in. Don't forget this, 3 out and 2 in. Okay. Webbing, or it's a cardiac glycoside which will inhibit to the binding site of the potassium. This is high yield. That is, you have a cardiac glycoside which will bind to the potassium site. So what would happen is that the potassium, they cannot enter. That's that. And then we have digoxin and digitoxin, which will inhibit the the potassium ATPase, which leads to indirect inhibition of sodium calcium exchanger, increase in calcium concentration, and cardiac contractility. We talked about this in details in the cardiac pharmacology. That is, if we block the sodium potassium ATPase, so sodium cannot come in. As a result, sodium and calcium exchanger is activated, which results in increase of serum calcium. As a result, sodium comes in and calcium goes out, and as a result, you have increased cardiac contractility. Okay, so that's that. We'll talk about this in details in CVS once again, and for all the students who have done CVS, then you guys know exactly what we're talking about. So sodium potassium pump as a whole is very high yield, but this is not, we are not unaware of this. Okay, this is very well discussed. So we all know what a sodium potassium pump is. So this is what a sodium potassium pump. Okay. Is everyone aware about this? Sodium potassium pump. Break. Anyone want a break? Does everyone want a break? Okay. Okay, before we leave for the break, I wanted to ask you about the lectures, with genetics. Are they helpful or not helpful? Are they helping you guys out or not helping you guys out? Or are they confusing you guys more? Okay. So what would happen is, let me tell you what would happen. After you guys are done with first aid with us, you guys know that you have to read first aid again. Am I correct? You guys have to read first aid once again, right? Obviously. So our goal is to make sure that you fall into those group of students who talk about finishing first aid in 2 weeks or 3 weeks. So what you guys can do is, after you guys are done reading with us, okay, maybe you guys can form groups within yourselves. Okay, we have the step 1 discussion group on Facebook. If you guys want to form a small group, for example, of 2 or 3 students, I personally feel like anything more than 2 is a party. Okay, well, what does that mean? That is, if there's 3 students, then 3 students in one group, then there's a high possibility things can go very wrong, because it's not very likely that all 3 of them can be present and have the same mindset at the same time. So it's better to form a study group of 2. So if anyone wants to start the revision process, then I suggest you guys find a study partner amongst yourself, okay, and start with the discussions. That's every day after we are done with our lectures. Maybe, for example, student A and student B, they have a study group. And every day after they're done with our lecture, they can have their own half an hour to 45 minutes where student A and student B can ask each other questions, repeated questions, fireball questions, and they can answer the questions very fast or not, and then they would know where they are lacking. Then they can read those parts once again. Okay, so what that will do is that will allow you to revise first aid very quickly. So this is just a suggestion. I'm not sure if that would be helpful, but if that is, then that's what you guys can do. Use the discussion group on the Facebook to find a study partner. Okay, so with that being said, we'll take a break for 10 to 15 minutes. Thank you so much for letting us know that our lectures are helping you guys out. We are very grateful to have students like you guys who are extremely intelligent and who can catch everything so quickly. And with that being said, I hope you guys take a good break for 10 to 15 minutes. Come back, because we are going to finish the cellular portions of biochemistry very soon. Okay, I'll see you guys in 10 minutes. Okay, is everyone back from their break? Okay. How do we feel about the topics that we are learning today? Are we understanding the topics? Yes, sir. That's my first question. And easy. It's not easy. USMLE genetics is actually pretty tough. I mean, this is also the USMLE biochemistry is actually pretty tough. But to be honest, could we succeed in telling you exactly where the questions will come from? And did you guys understand what to target the next time when you guys first date? Okay, very well. So if that's what it is, then let's go and talk about the second portion of this thing. Okay, so let's begin. First and foremost, so we're going to talk about the collagens. Collagens, we have type 1, type 2, type 3, type 4. And what we are going to do is we're going to take a blank page and then we will tell you exactly how you can understand all the different types of collagen. So first of all, the collagen is the most abundant amount of, it's basically the most abundant protein in the body. Okay, it is extensively modified, meaning that after it's formed, it's modified extensively by post-translational modifications. It's modified by post-translational modifications, which organizes and strengthens the extracellular matrix. So what happens is over there, after the protein is formed, that is a protein for collagen, you have exceptional post-translational modification, which modifies it and makes it tough so that it can withstand all the pressures a collagen is supposed to withstand. So first of all, how many types of collagen are there? We have 4 types. This is number 1, type 1 collagen. I'm not sure if you guys know this, but type 1 is present in bone. Bone, B-O-N-E. Okay, primarily type 1 is present in bone. So 1 for bone. Okay, along with bone, they have, along with bone, do you know what, what's on top of the bone, right? You have the bones, right? You have the bones. Then you have the muscle tendons. Am I correct? You have the muscle tendons like this. Not the muscle. Make sure it's not the muscle. It's the muscle tendon. On top of the muscle tendon, you have the skin. So, along with this, they are also present in dentine. Okay, dentine. Dentine. This is tooth. So the dental structure, that is the dentine, is a tough collagen that overlaps the dental structure. So dentine, and then over there in the muscles, you have the muscle fascia. And there's another thing. That is over here. You have cornea, right? And another one is in the stages of wound repair, type 1 is, if type 1 is present in the bone, is type 1 a strong type of collagen or a weaker type of collagen? I need some fast answers. It's a strong collagen if it's present in the bone because bone is strong, right? So if in stages of wound repair, in the early stages, do we have a weak collagen or do we have a strong collagen in the early stage of wound repair? Weak. But what happens in the late stage of wound repair? Do we have a weak collagen or do we have a strong? So what is the type of collagen in the late stage of wound repair? What is the type of collagen in the late stage of wound repair? The type of collagen in the late stage of wound repair is type 1. So late wound repair. Okay. These are all the places where type 1 is present. Okay. Have you guys ever heard of memory championships? Do you guys know what memory championships are? Memory championships. Memory championship. Memory championship is basically a tournament where you have players who are tested and compete against another players on who can memorize things faster. Okay. And the easiest way, if you watch a document on memorizing championships, then they will tell you that the easiest way that they memorize stuffs is by making a drawing or keeping something in mind. Because how hard is it to remember a movie? How hard is it? It's not hard at all. But how hard is it to remember a book? If you read a book, it's harder. But if you look at a movie, it's easier. Same way, if you have these drawings, then you can easily remember that type 1 is present in the bone. 1 for bone. If you can draw a bone, then you have the tendons, fascia, skin, then you draw another teeth, dentine, cornea, and late stage of wound repair. So this will be very easy. So am I clear about how you can remember the type 1 collagens? Because this is extremely high yield. This is extremely high yield. Okay. Okay. So that's that. This is type 1. Okay. Next one is type 2. Type 2 is very easy to understand because the word 2, okay, so type 2 is cartilage, meaning type 2 is present in cartilage or cartilage. Cartilage. Along with cartilage, we also have two other structures. That is, in the spinal cords, right? You have the discs, right? You have the intervertebral discs. In the intervertebral disc, you have two things, annulus fibrosus and nucleus pulposus, right? The middle one, nucleus pulposus, this is also type 2. And in the eyes, okay, in the eyes, you have the aqueous humor and then you have the vitreous body. The vitreous humor or the vitreous body is also type 2. So these are the three things which are from the type 2. Are we clear about this? Okay, so that's that. These are the type 2s. Okay. Nucleus pulposus is the part of the cartilage, which is intervertebral disc, the inner portion, nucleus pulposus. Okay. Next one. Next one is type 3. Type 3. Type 3 is comparatively, okay, so type 3. Type 3 is present in, okay, skin, then type 3 is present in blood vessels, and then type 3 is present in uterus and fetal tissues. Uterus and the baby over here, and the baby in the uterus. So uterus and the
fetal tissues. And if type 1 is a stronger collagen, type 3 is a weaker collagen. So in which stage of wound repair should type 3 be there? Early stage or late stage? Early stage or late stage? Early stage. Type 4 is in early wound repair. Okay, so this is type 3. This is type 3. Okay. Next one is type 4. Next one is type 4. Type 4. Okay, next one is type 4. Type 4 is present in, okay, we have type 4. Type 4 is under the floor. Under the floor for type 4. Okay, under the floor for type 4, meaning everything that is under the floor, especially meaning basement membranes, as basement membranes and lens. Basement membrane and lens. So another thing is, if we want to draw, if we want to draw the eyes, okay, if we want to draw the eyes, okay, then this portion, that is the cornea, is type 1, lens is type 4, vitreous body is type 3, vitreous body is type 3. Okay, so that's that. So these are all the cartilages. Once again, type 1 is present in the bone, skin, tendon, fascia, dentin, cornea, late wound repair. Type 2 is cartilage, nucleus pulposus, vitreous body. Type 3, skin, blood vessels, uterus and fetal tissues, and early stage of wound repair. Type 4 is under the floor, so basement membrane and lens. And then eyes are basically cornea is type 1, lens is type 4, vitreous body is type 3. Okay, can I give you guys one minute, 2 minutes to learn this? Okay, 2 minutes, and then I'll ask you which cartilages are present in which portions, and then we'll move on to the next one. Okay, 2 minutes on the clock. Vitreous body is type 2. Okay. Okay. So 2 minutes on the clock. It's 11:59. I'll ask you at 12:01. Okay. Type 1. So type 1 cartilages. Where can we find type 1 cartilages? Fast answers, please. Type 1 cartilages. Bone, then else. Bone, skin, tendon, dentin, fascia. Good. And wound. Which is wound repair? Okay. Late stage of wound. Very good. Next one. Next one is okay. Next one is type 2. Where can we find type 2? Cartilage and vitreous body and nucleus pulposus. Very good. Okay, nucleus pulposus. Do I have a broken voice? The voice being is the voice broken? Okay, let me come back. Is it broken? How about now? How about now? Okay. What are the structures arising from type 3 cartilage? From type 3? Type 3. Skin, blood vessels, uterus and fetal structures, and which stage of wound, early or late? Early wound. Okay, very good. Next one is type 4. Where do we find type 4? Type 4 under the floor, meaning basement and left. Okay, now let's go back to our text. So we have discussed all of this. Okay, now type 1, the disease, if there is decreased production of type 1, the disease you can come up with is Osteogenesis Imperfecta. That is in type 1. In type 3, if it's not work, if there's a vascular defect of type 3, because type 3 is responsible for blood vessels, the disease you come up with is Ehlers-Danlos Syndrome. Ehlers-Danlos Syndrome. And type 4 is under the floor. Obviously, if you have a type 4 defect, you can have Alport syndrome, or if you have autoantibodies which are targeting a base membrane, then there is another disease known as Goodpasture syndrome. We have talked about this before, so we won't be talking about this again, but we will talk about Osteogenesis Imperfecta and Ehlers-Danlos syndrome in 1 second. Before we do that, let's talk about the structure synthesis of collagen. Okay, the collagen structure and collagen synthesis basically. Okay. So collagen structures and collagen synthesis, let's go step by step. Okay, let's go step by step. So first of all, in the first step, that is in the synthesis step, you have translations of collagen alpha chains, that is pre-pro collagen. Pre-pro collagen are the alpha chains, and they usually contain glycine with either proline or lysine. But collagen is 1/3 lysine, and glycine content of collagen is less variable than that of lysine and proline. So if anyone asks you which amino acid in the collagen is the most highest, and what should be your answer? This is a very old question. Should be your answer? Very good. The answer is glycine. So write it down over here that glycine is the most abundant collagen in the, is the most abundant amino acid in the collagen. Okay. Hydroxyproline is used for lab quantification of collagen. So if you want to test how much collagen there is in the lab, what you want to test is another thing, that is hydroxyproline. Hydroxyproline, that is the hydroxylated portion of the proline. So what happens is alpha pre-pro collagen, they're usually glycine with either lysine or proline. Okay. Then the next step is hydroxylation. What is hydroxylation? Hydroxylation is this step over here. As you can see, that there's addition of hydroxyl groups. There's addition of hydroxyl groups to either the proline or to the lysine residues. And it's in this step that in the process of hydroxylation, you need vitamin C or ascorbic acid. You need vitamin C or ascorbic acid. So in a patient who does not have vitamin C, this disease is known as scurvy. So can we say that they have weak collagens? Yes or no? Weak collagen. Yes, okay. So at which step, let me make a note over here, okay? Let me make a note over here. Okay, I need you guys to make this note too. It's, okay, the note is name of the disease and step affected, step of collagen affected. Okay, the name of the disease, first disease is scurvy. And the affected step is hydroxylation. Okay, second one. Next one, after hydroxylation, you have glycosylation, meaning after you add the hydroxyl groups, what you want to add is you want to glycosylate or add the glucose or add the glycosyl groups of pro-alpha chain and hydroxylysine residues and form a pro-collagen via hydrogen and disulfide bond. So what you do is this one segment over here, if this has to loop and overlap and form this complex segment, right? If you add glucose is a pretty the way I like to remember is glucose is sticky. You know, like how when you want to stick two things together, if there's sugar over there, sugar helps it stick together. So the next step after you add hydroxylation, there is glycosylation. That is, you add the glucose, which makes it sticky so that they can form the triple helix. So glycosylation is basically the addition of the glycosyl groups of the pro-alpha chain and hydroxylysine residues, and this will form a pro-collagen. And this will help in the triple helix formation. Once again, whenever you add glucose or any other sugary substance, this adds as a sticky thing, which allows the thing to make a more complex structure. So if you want to add and form a triple helix, then you add the glucose. This will make it sticky. That is the best way you can remember this. If you do not have proper functioning of glycosylation, or this addition of glucose is not happening in a proper way, then the triple helix is not formed. And as a result, you have a very important disease. This is known as Osteogenesis Imperfecta. So the next disease is Osteogenesis Imperfecta. And which stage is this affecting? The stage that this is affecting is glycosylation. The stage affected is glycosylation. As a result, you have Osteogenesis Imperfecta. And glycosylation and triple helix is not formed. So glycosylation and triple helix formation. This is Osteogenesis Imperfecta. Next one, after you have formation of the triple helix, this is what happens is this is transported to the Golgi body. From the Golgi body, it is transported out, right? Via the process of exocytosis. So exocytosis occurs where the procollagen is transported out. After it is transported out, there is breakdown of the disulfide rich terminal ends of the procollagens. So as a result, when you break down the disulfide groups, this becomes more insoluble. If there is presence of disulfide group, this is more soluble. If you cleave out the disulfide groups, so cleavage of procollagen, C and N terminal ends, and you get rid of the disulfide groups, this results in insoluble collagen. We want the collagen to be insoluble because if the collagen was soluble, then it would be weak. Since it's insoluble, that's how it keeps the things in places, right? So you cleave the C and N terminal end, and you make the collagen insoluble. After that, you have the last process, that is cross-linking. Cross-linking is basically reinforcement of many staggered tropocollagens. So this is one collagen where there's insoluble collagen, and these collagens are cross-linked by covalent lysine hydroxylysine cross-linkage. So one of them is added to another one by a covalent bond. That is lysine with hydroxylysine. They form a covalent bond. And in order for this bond to form, a very important enzyme is required, and that enzyme is known as lysyl oxidase. If you have a problem with this enzyme and cross-linking does not happen, you have a very important disease known as Menkes disease or Menkes disease. So over here, the next one is Menkes disease. Menkes disease. Menkes disease, the stage affected is cross-linking. Okay, the stage affected is cross-linking. So for scurvy, hydroxylation. For osteogenesis imperfecta, glycosylation. For Menkes disease, cross-linking. Okay. Are we clear about this? No? Okay, please make the notes because you guys will be asked questions regarding this one. In your USMLE, they will ask you what is the name of the step that is affected, and the step for scurvy is hydroxylation. If you have a note, it will be easier for you to read and review faster. Okay. Yes, it's enough to know only this for USMLE. Okay. Okay. So that's that. Okay, which stage is affected in scurvy? Affected in scurvy. Okay, which is affected in Osteogenesis Imperfecta? Okay, which stage is affected in Menkes disease? Very good. Okay, so that's that. Is everyone clear and confident about the collagens? Okay. Let's move to the next one. That is, we want to discuss in details about osteogenesis imperfecta. Osteogenesis imperfecta is the problem where you do not have proper glycosylation and triple helix formation. As a result, the collagens are extremely weak. So you have weak collagens. The genetic bone disorder is caused by a variety of gene defects, most commonly COL1A1 and COL1A2. Okay, I'm going to make another table over here for the genes of the collagens. Okay. Okay. Disease and genes, because the genes are important. Okay. First one that we want to study is osteogenesis imperfecta. The genes are COL1A1 and COL1A2. Okay, so COL1A1 and COL1A2. That's what we want to discuss. And what happens in over here is that you have a bone disorder which is caused by COL1A1 and COL1A2. The most common form is autosomal dominant. So this is an autosomal dominant defect with decreased production of all types of type 1. With decreased production of otherwise normal type of type 1. So type 1 production is decreased. As a result, we talked about this before, that type 1 is responsible for the bones, right? So if you have decreased formation of type 1, you have weak bones. The manifestations are multiple fractures and bone deformities after. So you can understand that these patients, since they have weak bones, when you hit them, will they have a faster rate of fractures or not? Yes or no? Will they have fractures? No. Yes. Also, another one is if these patients, they have decreased type 1. Type 1 is also present in the cornea. Am I correct? Yes or no? Yes or no? If you have translucent or very easy to see through cornea, then is it possible to view the underlying veins in the cornea? I mean, like in the eye, the choroidal veins. Yes. So these patients, they also have blue eyes. As you can see, blue eyes, blue sclera, because they have translucent connective tissue over the choroidal vein. So you can easily visualize them. Next one is type 1 collagen. It's used for the formation of the dentines. Do you guys remember dentine? Yes. So in type 1 collagen defect, will they have strong teeth or weak teeth or poor dentures? Weak. So they have dentures. They have some forms of tooth abnormalities, including opalescent teeth that wears off easily due to lack of dentin. Okay. And another one is you have malleus, incus, and stapes, which are also bones. So the type 1 is affected. So you will have conducting hair loss, because there will be a conducting type of hearing loss, because malleus, incus, and stapes are also not working properly. So the ossicles are abnormal. As of that, you will have that. you will have conducting type of hair loss. So once again, what are the clinical features? First of all, multiple fractures, blue sclera, then conducting type of hair loss, and poor dentures. Poor dentures. Are we clear about this? And what is the gene that affected? The gene that is affected is COL1A1, and COL2. That's that. Okay. This may be confused with child abuse, right? And the treatment is bisphosphonates. We give the patients bisphosphonates, and bisphosphonate will prevent, will decrease the risk of fractures. Okay, bisphosphonate will prevent osteoclastic activities. So bisphosphonates will decrease fracture risks. This is not high yield. You're not asked about the treatment. You're asked more about the genes and the clinical features. Clinical features are the question, and the answer is usually the diagnosis or the genes. Okay, next one. Next one is Ehlers-Danlos Syndrome. Ehlers-Danlos Syndrome. Okay. First of all, is anyone confident to name all the clinical features of Osteogenesis Imperfecta before we move forward? Is there anyone who can? Okay. Who is confident? Who is confident? Okay. Dr. Iman, would you be kind enough to name all the clinical features of Osteogenesis Imperfecta? In the chat box or in the voice box, whichever you prefer. Fast, please. Okay, how about now?
Yes. Blue sclera.
Blue sclera.
Yes.
Then?
Football fractures.
Fractures. Then?
Hearing loss. Conductive.
Poor dentures. Then?
And teeth problem.
What?
Teeth.
Hearing loss. Okay, good. Very good. So those are the clinical features of Osteogenesis Imperfecta. Very good. Next one is Ehlers-Danlos Syndrome. Ehlers-Danlos Syndrome is faulty collagen synthesis, which can cause hyperextensible skin, hypermobile joints, and tendency to bleed. Do you guys remember seeing multiple talent shows where you have certain dancers who can twist and turn their arms, and they can make their feet up really high and then bend in a weird way, right? They're very bendy, they're very stretchable, right? So these guys, high possibility that they have Ehlers-Danlos. They're hyperextensible skin and hypermobile joints. There are multiple types of Ehlers-Danlos syndrome. Inheritance and severity will vary. They can be autosomal dominant or autosomal recessive. That is not high yield, but there is joint dislocation. There is joint dislocation, berry aneurysm, aortic aneurysm, and organ ruptures. So these patients, they can have aortic aneurysm, berry aneurysm, and dislocations. So watch out for questions from cardiovascular system for Ehlers-Danlos syndrome. Okay. So, and also from the neurology, because they can present with subarachnoid hemorrhage, those type of questions, aortic aneurysmal questions. So watch out for those questions. The hypermobile type is most common. And the mutation is, there are two types. There's classical and vascular. So for the classical type, you have, so Ehlers-Danlos syndrome, classic. So for the classical one, the mutation is in COL5A1 and COL5A2. And for Ehlers-Danlos syndrome vascular type, you have the mutation in COL3A1. COL3A1. Okay, COL3A1. So Ehlers-Danlos syndrome, the classical type is COL5A1. This is COL5A2. The vascular type is COL3A1. The vascular type means that the blood vessels are very, very fragile. So are the muscles and organs, and are more prone to rupture, especially if you have a female who is pregnant, then high possibility the uterus might rupture because the structures are not very high yield. I mean, the structures are not very strong. That's that. The mutation is in type 3 collagen, so COL3. And this is in type 5 collagen. Although over here, they have not mentioned type 5, but apparently there is another type. So that's that. So there's not really much to think about this, then only try to remember this. So the patients have hypermobile, hyperextensible skin, laxative joints, berry aneurysm, aortic aneurysms, chance of uterine rupture, right? So these things. If it's a vascular type, it's called 3A1. If it's a classical type, it's called 5A1 or called 5A2. Okay. Is that clear about Ehlers-Danlos syndrome? Is everyone clear about Ehlers-Danlos syndrome? Okay. Okay, very good. Next one is why is there a tendency to bleed? There's a tendency to bleed because the blood vessels are very fragile. Okay, because you have type 3 collagens, which are responsible for the blood vessels. And since they are fragile, hypertension can result in vascular bleeding. Okay. Okay, next one. Next one is Menkes disease. We have talked about this, that Menkes disease occurs in the last stage, that is in cross-linking. You need lysyl oxidase to form the links between these type of the covalent bond, that is hydroxylysine and lysine residues. And lysyl oxidase needs copper, right? Needs copper. The Menkes disease is actually an X-linked recessive connective tissue disorder, which is characterized by impaired copper absorption and transport due to effective Menkes protein. Over here, the gene is ATP7A. We know that in Wilson's disease, the gene is ATP7B on chromosome number 13. And over here, you have Menkes disease. You have Menkes disease, which is ATP7A. 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 And the copper, this affects the copper absorption. As a result, if copper is not there, lysyl oxidase cannot really form or catalyze the bond between lysine and hydroxylysine residues, and that's why you have the Menkes disease. So this affects the last stage, cross-linking, leads to decreased activity of lysyl oxidase and defective collagen. How can you identify Menkes disease? These patients will have abnormal hair, growth retardation, hypotonia, and cerebral aneurysms, increased risk for cerebral aneurysm. So this is basically, the questions are, fortunately, since the clinical features are not very particular to anything, they will direct the questions to and tell you that these patients, they have some sort of a copper abnormality. And that's when you have to realize they're talking about Menkes disease. And then you can identify that Menkes disease is happening because of your problem in the cross-linking stage. This is basically the question which they will keep on asking you from time to time, that which stage is affected, and the stage is cross-linking. That is the 6th stage. Which enzymes are affected? The enzymes are lysyl oxidase. Lysyl oxidase. Okay. Are we clear about this? Menkes disease. Menkes disease, once again, on the last stage, in order for the bonds to form between lysine and hydroxylysine residues, you need the enzyme lysyl oxidase, which has to work. And this has to work in the presence of copper. So we have Menkes disease, which is a copper absorption defect, where due to the defective gene ATP7A, the copper is not absorbed. And as a result, the lysyl oxidase, it does not function properly, and you have defective collagens. Okay. Are we clear about this? Can we move on to the next page? Okay. Let's move on to the next page. That is elastin. What is elastin? Elastin is basically, obviously it's a protein. So it's a stretchy protein within the skin, lungs, arteries, elastic ligaments, vocal cords, ligamenta flava, right? And ligamenta flava. So elastin is in everything which needs to dilate, right? I mean, dilate in the sense that which needs to move up and down. So skin, elastic, large arteries, elastic ligaments. The locations of the elastins are not high yield. I'm going to tell you why. Because all the regions of the bodies where collagen is in low amounts, elastin is in high amounts, right? So ligaments are an elastic structure, meaning it's stretchable. So if you stretch it, it will come back to its original position. So the elastin, they are rich in non-hydroxylated proline, glycine, and lysine residues. Okay, so over there, you also had glycine, proline, and lysine, which was hydroxylated. But in elastin, the difference between collagen and elastin is in collagen, you have hydroxylation of the glycine, proline, and lysine. In elastin, you do not have the hydroxylation. So they are non-hydroxylated. As a result, since they are non-hydroxylated, the tropoelastin, this one, can fold with the help of another protein known as fibrillin. So they can fold like this, tropoelastin with fibrillin scaffolding. The cross-linking, that is the linking between one elastin and another elastin, takes place extracellularly, and it gives elastin its elastic properties. So this cross-linking, right, this is responsible for giving the elastin its elastic properties. That is, it's stretchable. If it can stretch, it will come back to its original position. Now, elastin can be broken down by a protease enzyme known as elastase. We know who is responsible for secreting the elastase, that is neutrophils, right? Which is normally inhibited by alpha-1 antitrypsin. So you have a protease, and then you have antiprotease. If you have antiprotease deficiency or alpha-1 antitrypsin deficiency, this results in unopposed elastase activity, and this can cause COPD. We talked about this before. Also, what happens with aging is when a person ages, that is, they get older, they have decreased collagen and elastin synthesis. As a result, in old people, you will see that their skin becomes less elastic, right? And then you have decreased dermal collagen and elastin and decreased synthesis of collagen fibers. So the collagen fibers are also synthesized in low amount. But the only thing that remains normal is cross-linking. Cross-linking remains normal. Okay, so let's do a quick revision. Elastin is nothing but a stretchy protein. That's number one. Number two, elastin is present in all the places where connective tissue is not there. That's number two. Number three, elastin is non-hydroxylated glycine, proline, and lysine versus the collagen, which is hydroxylated. Number four, elastin is covered by a fibrillin structure, which helps it form into a scaffolding structure like this. Number five is there is cross-linking between the elastin, which helps and gives the elastin its elastic structure. Number six is the cross-linking stays normal in aging, but everything else decreases in aging. That's that. And number seven is elastin can be broken down by elastase, but alpha-1 antitrypsin works against elastase and prevents the overactivity of elastase. So in a patient with alpha-1 antitrypsin deficiency, you can get COPD. We talked about before. Is everyone clear about elastin? What are the questions you will receive from elastin? The questions you will receive from elastin is fibrillin. If there's a question regarding fibrillin, you have to realize that they're talking about elastin. Next one, if they are talking about non-hydroxylated proline and glycine, they are talking about elastin. Okay. If they are talking, number three, if they are talking about protein, which can come back to its original size, then we're talking about elastin. So the only questions you will receive from elastin are identification, rich in non-hydroxylated proline and glycine residues. This is extremely high yield. Underline this. Then fibrillin scaffolding is extremely high yield because they're present only in elastin. And then obviously trypsin and alpha-1 antitrypsin, they're also very high yield, right? So that's that. So these are the three things that you have to understand and remember. Are we clear? Okay. Okay. Next one. Next one is Marfan syndrome. Marfan syndrome. Have we heard about Marfan syndrome before? Yes or no? Right, very good. So Marfan syndrome is a connective tissue disorder which results in abnormal fibrillin. So there's this, so do you guys remember we talked about fibrillin scaffolding, which helps it maintains the elastin its shape. So if you have abnormal fibrillin, then you have a connective tissue disorder called Marfan syndrome. It's an autosomal dominant connective tissue disorder, which will affect the skeleton, heart, and eyes. Okay. What would happen is over here, you have a mutation on chromosome number 15. So Marfan F for 15 for FBN1. So the last connective tissue is Marfan syndrome, and the gene is FBN1. FBN1. Okay. FBN1. So over here, you have FBN1 mutations on chromosome number 15, which results in defective fibrillin. And what happens is when you have defective fibrillin, the elastin, they have a more ability to stretch out. That's why these people, they have this elongated, tall appearance with extremely stretchy joints, right? So once again, they have hypermobile joints, long fingers, toes, right? And in the heart, what they have is they have cystic medial necrosis of the aorta. Okay, but more important is aortic root aneurysm. These patients are more prone to aortic root aneurysm. Aortic root aneurysm, once again, is the balloon-like dilatation near the aortic root. And in cases of hypertension or trauma, this can rupture, making the patient go in shock. So that's that. And these patients, they also have mitral valve prolapse. And another one is lens subluxation. I wanted to talk about this. Lens subluxation. Lens subluxation can occur in two ways, and this will help you in identify whether you are dealing with Marfan syndrome or homocystinuria. If this is the lens, if this is the eyes, right? If this is the lens subluxation is basically dislocation of the lens. If the lens typically move, so this is the nasal, this is temporal. If the lens move upward and temporally, this is Marfan syndrome. Lens dislocation in this direction is an identification of Marfan syndrome. If the lens move downwards and nasally, this is an indication of homocystinuria. I need you guys to draw this diagram over here in this portion. Draw this diagram over in this portion so that you can identify this, because this will be a very easy mechanism by where you can identify whether you're dealing with homocystinuria or Marfan syndrome. And that being said, we are done with the cellular portion of biochemistry. Okay, has everyone understood this? Okay, I'll give you guys exactly 2 minutes. I'll give you guys exactly 2 minutes. I need you guys to learn this right now, because this is something which you have to memorize. Unfortunately, there's not much to understand. I need you guys to learn this right now. I'll ask you guys questions in 2 minutes. Okay, in 2 minutes. Okay, 2 minutes on the clock. I'll ask you guys questions at 12:36. Okay. So let's talk about the genes now. Okay, 2 minutes are done. 2 minutes is over. So first of all, for first disease that we talked about, let's talk about Ehlers-Danlos Syndrome, the vascular type. What is the gene What is the genetic defect in Ehlers-Danlos syndrome? Okay, what is the defect in the type? What is the defect in the class type? All 5A1 and next one is Osteogenesis Imperfecta. What is the defect? Okay. The defect in Marfan syndrome? Okay, good, very good. And then what is the defect in Menkes disease? Okay. Okay. How confident are we? Are we confident that we can we can give the correct answers every time? Elastin is basically the glycine, proline, and lysine residues which are non-hydroxylated. Since they are non-hydroxylated, they need to be that is elastin, and then what happens is that the elastin is covered by another cover known as fibrillin, which maintains the structure of the elastin. So if the fibrillin is not working properly, then the elastins have the more ability to stretch out, and this results in Marfan syndrome. So that's that. That is all you need to know from elastin. Okay. Okay. You are welcome. So can we keep our first aid reading up to here, and then we can start with the questions. Okay. So tomorrow we will begin with the lab techniques. Okay, lab techniques, these ones. Okay. Okay, so do we have any student who does not want to do classes on Christmas? Or what about Christmas? Do you guys need a break on Christmas, or is it? Okay. Me, I can't. Okay. 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 We will do is we will come to a decision. Okay. Okay. So I know that it's a big holiday. It's one of the biggest holidays, and we were planning on not giving any breaks. But to be honest, at this point, we're still not sure whether the classes will happen tomorrow or not. We will confirm it for you by tonight. Okay. We will confirm it for you, right? And if the classes don't take place, then obviously we'll try to make it up. So we will make up the classes next week. Okay. So that's that. The thing is, if we do not do classes tomorrow, then we have to do an extra class next week. So that's okay. So we will let you know whether we plan on giving the break or not accordingly. Okay. So let's do something. Let's take a 5-minute off, and then let's come back and begin with the questions. Okay. Okay. Okay, is everyone ready to begin the genetic questions from plan? Are we ready? Okay, great. So let's begin with the questions. Let me share my screen. First and foremost, I will use the chat box to answer the questions. And if there is any student who needs an answer, who has the answer to the question, just tell me you're ready, and then we can provide the answers. Okay. Thank you so much for doing the questions. I know a lot of the students, they usually leave by this point, but it's important you do the questions so that you understand how to attack the questions. Okay. Okay. Some of the things over here, we have not studied them before, but let me see if we can get an answer out of you guys. Because hopefully, well, let's say, I mean, not hopefully, I mean, there's a chance that you guys will receive questions in your actual exams, which you have never studied before. So let's see if you have the ability to break some of the questions down. And we're also here to help you understand why the answer is the right answer. Okay. So you have 90 seconds on the clock for this question over here. The first thing that you have to do is read the last line of the question. And if you have the answer, then please say you're ready. We'll use the chat box. Okay. So, okay, let's begin. First of all, the last question is, his blood glucose level is now 60%. Let's start from over here. A 30-year-old man has been fasting for religious reasons. His blood glucose level is about 60% of its normal value, but he does not feel lightheaded because his brain has reduced its need for serum glucose by using which of the following substances as an alternative source of energy? This is actually pretty easy. Anyone is ready? Please tell me you're ready. Okay. Is everyone ready? Let's provide the answers. What are your answers? C. C, basically beta hydroxybutyrate, which is, what is beta hydroxybutyrate? It's a ketone. It's a ketone. In terms of low glucose, the body, the brain especially, will use ketone. Which other cell of the body will use ketone body for energy in terms of fasting? Anyone? Okay. I'll leave this question for later. I'll make you guys understand this later. So that was the first question. Let's move on to the second one. Okay, let's move on to the second one. The mutation is most likely in a gene encoding which of the following? A lethal mutation occurs in a bacteria, rendering it incapable of replicating its chromosome. Because of its mutation, DNA synthesis produces many short fragments of DNA that have RNA sequences at their 5' ends. They have RNA sequences at their 5' ends. The mutation is in which one? So take your time, let me know when you're ready, and just tell me you're ready, don't provide the answer. Okay. What is the answer? Okay. C. Why is everyone writing C over here? Okay. Why is the answer C? Most of you guys answered C. Okay. Let's read the question. The mutation occurs when the DNA synthesis produces many short fragments of DNA that have RNA sequences at their 5' ends. What is the name of the RNA sequence? What are these RNA sequences? First of all, what are the names of the RNA sequences? Primer. Which gene or which enzyme is responsible for removing the primers? Removing of the primers, not the addition of the primers. Primase is responsible for addition of the primers, not removal of the primers. Enzyme DNA1, exactly. DNA press 1 is not working properly, then there will be RNA primers added to the DNA. Okay, do you guys realize how easy this question is, but everyone gave the wrong answer because they did not understand what the question was asking you? The question told you that there's DNA with RNA attached. What is the name of the enzyme that is responsible for letting the primers go away? The enzyme is DNA polymerase 1. So this is a very good question. Yes or no? Is this a very good question? Yes. Okay. Okay, next one. Yes, primase will add, polymerase will, DNA polymerase 1 will remove it. Okay. Which of the following enzymes was most likely deficient in this child? A 2-year-old with a past medical history significant for mental retardation. Hepatosplenomegaly, foam cells in marrow. There's foam cells and neurological deficits. Okay. This is actually pretty easy. Okay. Wait, we have only 2 students who are ready. Well, which of the following enzymes was most likely deficient in this? Okay. Okay. This is a pretty easy disease. It is a lysosomal storage disorder. Well, what is the diagnosis of this patient? Diagnosis. What is the diagnosis? It's Niemann-Pick disease. It's not Gaucher's disease. It's not Gay-Sachs. There's no disease called, it's Tay-Sachs. Okay. Okay. No problem. No problem at all. Okay. So Tay-Sachs disease is not the one. It's Niemann-Pick disease. Niemann-Pick disease is this is Niemann-Pick disease. It's a disease where the patients have neurological deficits, hepatosplenomegaly, mental retardation, foam cells, right? And we will talk about those diseases. Do you know exactly how we will talk about this disease? Let me show you the best way we will talk about this disease, and you guys will never forget lysosomal storage disorders in your life. Okay, let me show you an example of how we will talk about this. And lysosomal storage disorders, they're coming very, we will talk about this very soon. Okay, I want to show you the picture that physio has, which is absolutely brilliant over here for the lysosomal storage disorders. Okay. Let me show you. These are the no, these are not the lyso ones. One second. There we go. And over here, you have the picture of Tay-Sachs and these. I am so excited to share this one with you. We will learn we will learn this next week, okay, by using the picture mnemonics, and you will never forget lysosomal storage disorders in your life. This picture of an old man who's picking his nose is for Niemann-Pick. The fact that he's holding he is picking his nose with a finger, we can say the words finger instead of finger, which is for sphingomyelinase deficiency. Over here, sphingomyelin is the accumulated substrate. So sphingomyelin. So finger for sphingon, finger for sphinger and sphingomyelin. Then you have this crane which has fallen down, which shows that this old man has neurological deficits. Then dog with liver spots for hepatomegaly, foaming at the dog's mouth for foam cells. And this is how you can remember all the clinical features of Niemann-Pick disease. Are we excited to do this? Yes or no? This is very, very exciting. And to be honest, what we will sure if we can next week, we had a student who said that is the voice breaking now, or how is the voice? Do I have a broken voice? Is the voice breaking? Okay. You had a student who said that what is the best way to learn about the vitamins? Let me show you what the best way to what the best way to learn about the vitamins are. Vitamins. There we go. You have all the picture mnemonics for all the vitamins. Every vitamin, vitamin B1, for example, you have the picture mnemonics for vitamin B1. See all this information over here for vitamin B1. It will be absolutely amazing. all you guys will have to do is look at cartoons for 3 to 4 hours, and you guys will be done reading and learning vitamins. Okay. Okay, so that's what it is. So that's what we're going to do. Let's go back to our, okay, let's go back to our question. So this is sphingomyelinase. Thank you and congratulations for giving the right answer. The diagnosis is Niemann-Pick disease, and the enzyme is sphingomyelinase. Okay, next one. Next one is this one over here. Okay. The missense mutation is most likely in the gene encoding an enzyme essential for which of the following functions? A 58-year-old woman is admitted to the hospital with fever and abdominal cramps. The symptoms began one day after attending a banquet. She was treated with ciprofloxacin, but her condition did not improve. PCR amplification and analysis of the gene region in the bacteria isolated from the patient revealed a missense mutation that conferred resistance to ciprofloxacin. Okay. In order to understand this, you have to understand the mechanism of action of ciprofloxacin. Does anyone know the mechanism of action of ciprofloxacin? What is the mechanism of action of ciprofloxacin? Okay. So which one of this is the right answer? Okay. DNA is important for DNA replication, as well as the right answer is A. The right answer is A. So good job. Very good. Right answer was A. That's that. Okay. Next one. Next one is this patient most likely has which of the following conditions? A 62-year-old man is prescribed a pharmaceutical agent that inhibits the activity of HMG-CoA reductase. Easy question. Which drug is given and for which condition? If you're ready, let me know you're ready. It's a very easy question. Okay. What? Nope. D is D. Because HMG-CoA reductase, for example, statins, there are statins. Atorvastatin, lostatin, rosuvastatin, right? They are for cholesterol. First step of cholesterol synthesis is HMG-CoA, so they are the cholesterol. Okay, good. Last one. Last one is this. Okay. A 5-year-old mentally retarded boy is brought to the city from a community. Then a careful history reveals mental retardation in a number of family members, especially the males. Physical examination is responsible. Physical examination is remarkable for a long with large jaws, lateral, and okay. So this is a pretty easy question. What is this? Very good. The answer is fragile syndrome. Okay. Can you guys just give me 1 second, please? Just give me Okay, so that's that. Okay, my apologies for taking that break. Thank you so much for doing the questions with absolute perfection. Has everyone understood the lecture for today? Has everyone lecture for today? Okay, good. Since tomorrow's a Christmas, we are done with a very big topic and more questions. Do you guys want to end the lecture over here right now? Okay. Right. So I hope you guys have a great Merry Christmas to one. I hope you guys have a great holiday. Let me know if there is anything you guys need or there's anything you need to learn. If you guys have a question, let me know. And if all, will see you guys tomorrow at 9. And tomorrow, okay, Merry Christmas, Dr. Lala. Hope you have a great day. Merry Christmas to you and your family. And we will tell everyone, we will send an email to everyone regarding the confirmation of tomorrow's lecture. Okay, so that's that. Okay, so we said thank you so much. Hope you guys have a great day. I'll see you guys tomorrow.